So we're going to talk now about epigenomic research. So Elin, are you around? Come over. You can introduce yourself, Elin, so please go ahead.
Thank you. Yes, I'm Elin Grundberg, and previously at McGill University in Canada, now relocated to the US. So I will talk with both hats on, McGill and the new institution at Children's Mercy Kansas City. First, thank you, Biobank, for this invitation. I'm very excited to be here. I see myself talking on behalf of many of you here that I've spoken to earlier today and on emails that are excited to see the possibilities of expanding the molecular profiling in UK Biobank, including epigenetic profiling. And I will show you some of our insight in doing this in large cohort, and hopefully we'll come up with strong proposal and convince funders that this is the next layer of information that we need in UK Biobank. So during the day today, I think we got an amazing overview about UK Biobank as a resource with deep phenotypes, but also on the genetic side, genotyping spanning now from array-based and now with Regeneron and other efforts with exome sequencing and as well as the whole genome sequencing. And similarly on the environmental lifestyle factor, how this is very well covered with a deep catalog of information. So what I will try to convince you is that we can use epigenetics to connect environmental with genetic factors and then learn phenotypic risk and disease biology. And I will talk about how we can use epigenetic changes or epigenetic variation to interpret or translate genetic variation into regulatory causal pathways. And that epigenetic variation might be superior to the previous transcriptomic variation that was popularly used in complex disease studies. But also show you that environmental lifestyle factors actually can impact our epigenome and cause epigenetic modifications, and that in a similar way can impact and identify causal and regulatory pathways for complex disease, and just touch upon the fact that compared to genetic factors and environmental factors, in terms of epigenetic changes, we can get a consequence of that based on the disease. So as probably many of you know, this is what we do when we do an epigenome-wide association study, very much similar to a genome-wide association study. We pick our study population of interest, either a case control or population-based, and we do epigenetic profiling, either dependent on funding or interest, targeted by array, or we do whole genome or at least next-generation sequencing-based, and then we do large-scale association studies to find regions of the epigenome in terms of marks that have an association to our disease of interest. I think many of you would agree with me that the most commonly used epigenetic trait in large-scale population-based studies is DNA methylation, and I will list 3 reasons why I think that's the trait to use. Obviously, we're dealing with very robust and reproducible assays. Most of them are based on bisulfite conversion of our DNA. And we do have automated solutions in place, meaning that we can do this now in large scale. And we both have microarray-based method as well as NGS that do this in a very accurate manner. Obviously, DNA methylation, it benefits from being a quantitative trait. We're mostly dealing with bulk tissue, so we're dealing with a distribution of 0 to 100% of our trait, which makes it easier than other epigenetic traits to do statistical analysis. But obviously, as we're dealing with a single base pair resolution variant, we do have up to 30 million of these sites that we can measure genome-wide. And also, compared to transcriptomic assays, epigenetic or DNA methylation is a stable trait. And we can use this actually as a predictive of regulatory element. So what have we learned in doing epigenome variation or DNA methylation in large scales based on assays? We started, and I have a long-standing collaboration with the Twins UK. So we did this a couple of years ago now, and we're interested to see if we do this genome-wide using whole genome bisulfite sequencing, how's the DNA methylation variation dependent across individuals, across tissue, and across regions of the genome. And it's really striking to see that DNA methylation variation is very region dependent. We do see very static patterns of DNA methylation in certain areas, such as the promoters, whereas the variability is really enriched in more distal regulatory elements as the enhancers. So we were not keen on using the larger scale, the targeted array at that time, the 450K array. We saw that it's really a bias or attainment with a large proportion of those CpGs being in promoters. Similarly, we noticed that where we have an association with a complex or a disease trait, these are also in a similar way as we've seen for genetic variants enriched in these enhancer element. Obviously, at that time, and still, whole genome bisulfite sequencing is costly, but also that we're dealing with a large proportion that's actually might not be very useful, as it's not variable across individuals. So we wanted to see how we can improve the profiling of DNA methylation by still restricting to certain areas. So we initiated a collaboration with Roche and Imbogen to follow the same procedure as a whole genome bisulfite sequencing, so a shotgun library, but instead of sequencing that full library, we target and enrich, similar as in an exome sequencing experiment, we enrich for regulatory elements, which we were interested in to do. And by doing so, we can do multiplexing, we can cut down the cost, and we can increase our coverage of the regions that we were interested. So we're designing these panels based on our tissue of interest. Obviously, blood is a tissue that we do have most samples available from. So we design panels based on regulatory regions specifically targeted for blood, so make sure that we're covering all regulatory elements in all, or at least the data that's available from different purified blood cells. And also we include other types of regions that we think is useful, for instance, the CpGs that have been added on other types of methods. The benefit from things with the genotype at the same time, so we made sure that we include also genetic variants, rare variants that we can then genotype at the same time. The bottom figures is basically showing what this looks like. Basically in the red bar showing the regions that we cover, and then oh, there we go. So basically this is what we cover, and this is when we're sequencing, and we can compare that with the whole genome. Obviously it's not fully covered, but compared with 450K, we do cover densely the regions that we're interested in. So by having sort of a full or dense coverage of regulatory element, we can also start to see how's the pattern of epigenetic association within an element. And we have started to detect very interesting features. This is showing a statistical significant association with a trait, and those that are in enhancers seems to preferably mapping at the midpoint of the enhancers. And if we then limit to promoters, the significant disease epigenetic variants have a preferable association towards the gene regions. So if we then mirror that and see what do we have available on arrays, we do see that there's an underrepresentation of that region, specifically on promoter, as which is the majority of the regions on this array. So we were kind of still interested to start to continue to do this dense coverage of the regulatory elements. So if we do that in disease studies, how do they look? We use cigarette smoking as one of sort of a proof of principle environmental factor, that epigenome or environmental factor actually changed our epigenome quite dramatically, but also learn what is the advantage of doing dense regulatory sequencing over scatters. So we obviously confirm this is the most famous chromosome 5 regions for smoking, the AHRR. That is the most reproducible and replicable finding in epigenome-wide association studies. But this is the enhancer that has been strongly associated with the activity and expression of this gene. But we also see 2 additional regulatory elements that show signals and being affected by smoking. But maybe most interesting is that we will look in this specific regions that we know has the strongest effect based on smoking, where the blue line is actually seeing if we sequence that element in higher depth, we do fine map the association and have a CPG with a much better predictable scores when we're trying to predict an individual's smoking status than the green one, which is the CPG that has been on the array-based. But also we're interesting to see if we have this preferentially mapping of epigenetic variants within regulatory element, is that useful? Does it tell us something? So we can zoom in and look at these associations in the midpoints as well as in the downstream of the promoter, and actually learn how the transcriptional regulation is linked to this. We find, in this case, is metabolic disease associations, and we see strong enrichment of transcription factors that are binding in these specific regions. And we can follow up and do additional thing. We can obviously do our replication, but we can link it to gene expression and then do similar thing. Instead of doing the regulatory element and transcription factor per se, we can see the downstream genes and do pathway analysis and actually see that similar pathways are coming up as our transcription factor binding analysis. And if we zoom in of one of these genes that has been strongly linked to this metabolic trait, we know it's the vascular endothelial growth factor. There's a genome-wide association study using the UK Biobank data here, strongly showing an association of downstream of the gene. We identify a regulatory element rather far upstream of the gene is a tissue-specific enhancer robustly associated with a metabolic trait that's relatively linked to the GWAS trait. And obviously, with the genetic information in these cohorts, we can integrate that and see that the association of epigenetic variation in this region is non-genetic. So that's basically potentially indicating that we have an additive effect of a locus that is relevant to this disease. Obviously, as I mentioned, a key feature of doing epigenetic or molecular profiling is to understand genetic susceptibility, and obviously we can do this too with DNA methylation, and as I said, even better than using gene expression. I take this example, which Nicole Soranzo, as we heard earlier today, published together with Tommy Pastinen at our institution a few years ago using the blueprint data, basically collecting large number of individuals with various different blood cells and different layers of molecular trait and trying to see what is the co-localization of a disease SNP versus molecular, and actually in that defined that gene expression only accounts for a minority of disease link, where the majority is actually epigenetic variation and specifically DNA methylation as a strong molecular trait explaining the link between a genetic variance and the disease outcome. Again, we can zoom in and take an example to try to link it to data from the UK Biobank. This is an interesting signals from the blood pressure consortium using the UK Biobank data. And similar here is a tissue-specific only occurring in blood tissue, a strong regulatory element with robust epigenetic variation of DNA methylation within the GDF7 gene. And compared to the previous example I took, if we integrate genetic information with this DNA methylation, we see perfectly nicely co-localizations where further integration with gene expression data actually can indicate that we're dealing with this gene that seems to be genetically modulated through epigenetic variation at this locus. Finally, as we're dealing with epigenetic tissue specificity and talking about blood tissue heterogeneity is a key thing to consider. It matters, it's important, and we need to account for that. I personally is less worried about this than previous year, basically since we know we can actually correct for this relatively well. Many of you have seen the vast amount of available packages, basically reference-free, reference-based, and mainly are supporting array-based data. What we actually do, we can actually do supervised prediction of cell counts in our next-generation sequencing-based data by basically identifying CpGs that are very specific to a blood cell type, and we can use publicly available data from up to 19 purified cell type, really be using stringent criteria and identify hundreds of CpGs that only active or hypomethylated in a cell type, and using those then to fetch the average methylation in whole blood data, we do see that they very well correlate with our lab test, and meaning that we actually can use this in statistical analysis in cohorts where blood counts are not available, and actually adjusting for this in a much more accurate manner than if we have the CBC test alone. So I'll summarize and try to highlight why I think epigenetics is an important and useful trait in large resources such as the UK Biobank. We do know that we can discover regulatory pathway that underlies complex trait and diseases. These can both be genetically driven, but with epigenetic, we can actually pick up environmental or lifestyle-driven effects. We believe that the high-resolution profiling of regulatory element has a great advantage, as it have this opportunity to add additional insight into regulatory pathways. DNA methylation really is a perfect molecular trait to annotate the putative functional consequences of genetic variants. And again, if we're doing very dense coverage of DNA methylation, we'll be able to cover the majority of these regulatory disturbances that non-coding genetic variants cause. And finally, the opportunity to sort of add epigenetic profiling really have the potential to allow us to build these combined risk scores, maybe including epigenetic variants as well, and for having prediction of later life outcomes. And obviously, we're really dependent on large cohorts and well-phenotyped studies in order to do that. And obviously, UK Biobank is the perfect tool for that. So I will end there, just highlight everyone that has been involved in our project, but specifically the UK Biobank team with Rory Collins and Paul Matthews, and both Jimmy Bell and Gemma that's here, that's been very supportive in discussing this, and hopefully we'll come up with a strong proposal and then convince funders that epigenetic profiling is the next trait to be assayed. Thank you.